send out links afterwards. Thank you, ma'am. So welcome everybody. We're just a couple of minutes after 12, so we're going to go ahead and get started, knowing there's a lot of things going on around us. Want to use your time wisely. So I'm Terry Cook. I'm the state director for the Nature Conservancy here in Tennessee. And for those of you who've been on or haven't been on, many of us just decided to hold a weekly brown bag to provide opportunities for us to learn about programs and projects that are going on across Tennessee from various partners. Just so you know, the different organizations that have been invited, we have the Wolf River Conservancy, who's joining us hopefully this week. We have Cumberland River Compact, Harpeth Conservancy, and the Tennessee River Gorge Trust. So a few of the partners that are doing great work across Tennessee, and just a unique way for us to get together. Many of us talked about not interacting enough and using this time to increase a little bit of interaction and knowledge between different organizations. Hopefully something positive will come out of that. One, we'll learn something, but there may be some new collaborations and partnerships that emerge from it. So today, Harpeth Conservancy is going to be presenting. Ryan Jackwood will be talking about Tennessee rivers and some of their water quality work. So Ryan, I'm going to turn it over to you.
Thanks, Terry. So I'm the Watershed Science and Restoration Director for the Harpeth Conservancy. I started about a year ago, a little over a year, I guess. And I came from the University of Toledo, where I was doing research on water quality in Lake Erie, and that's where I got my PhD. And noticed that Lake Erie and some of the Great Lakes are having a lot of similar water quality issues that are going on around the rest of the country. And so talked to Dory and came down here to work in the Tennessee area on water quality issues. So let's see if I can get, there we go. So I realize I'm preaching to the choir kind of with this, but I always think it's kind of important to explain why we should care about some of our natural resources at the beginning of presentations. And the southeastern US is a global aquatic biodiversity hotspot. These maps kind of show just hotspot areas for fish and freshwater turtles. And the southeast US is actually the third biodiverse area in the world. And so really the rivers in our backyards are really important for biodiversity and to keep, basically to conserve them. I also like to put up this map too, because it really kind of depicts the area and the size of various watersheds and then the number of fish species that we can find in them. And places like the Duck River and Paint Rock and these smaller watersheds down in the southeast have way more species of fish than some of our even huge watersheds out west. And so this area really is quite important for aquatic wildlife. We at the Harpeth Conservancy, we kind of use the Harpeth watershed as our model system, but really we're looking at watersheds all across the state of Tennessee and trying to explore water quality issues. But we're kind of focusing on the Harpeth River, and I'll talk about some of the work that we've done in the Harpeth and maybe how that work can kind of translate. But Harpeth has over 80 species of fish. It's a drinking water source for a lot of people. And what's kind of one of the more unique things about it is that it's highly recreated. We get over 500,000 people that recreate just in the Harpeth State Park areas alone. So that's not counting Franklin and some of the other city or metro parks. So as we kind of are looking at issues for rivers around Tennessee, it's kind of a lot of times we'll fall back to what would be what the Tennessee Department of Environment and Conservation considers impaired. And they list impairments like pathogens, nutrients, low dissolved oxygen, habitat loss. In this map on the left here, all of the streams and rivers that are in red are considered impaired by at least one of these impairments. And so it is our goal to kind of focus in on the Harpeth River and figure out, okay, how can we make sure that the water quality in the Harpeth River is in attainment, basically, that it doesn't have these impairments, and how can we improve the water quality so it meets the state standards. And so today I'm going to talk about two of these impairments and kind of some research that we've done and kind of some sampling that we've done. So we're going to talk about first, we're going to talk about nutrients, and then we're also going to talk about pathogens. And pathogens cover a huge wide range of microbacteria or bacteria, but we will kind of lump them all together and use E. coli as an indicator for pathogens. So first, nutrients. So nutrients, they can enter our waterways from a variety of different sources. These are kind of our two large categories when we consider how do nutrients actually contaminate rivers and streams. Point sources and non-point sources. Point sources, I oftentimes will tell my students that you can point to it. You can actually see where it's discharging or it's entering a body of water. And then non-point are these very diffuse sources of pollutants that can either slowly flow off of landscapes or run off, and generally driven by precipitation events. And so these would be two examples of an example of a point source and an example of a non-point source, how they're actually entering into our waterways. So end of pipe here on the left for a point source, and then our non-point source, that's going to be the edge of an agricultural field. And it just rained, and that runoff is eroding the sides of banks of rivers and flowing into those rivers. And that runoff is then carrying a lot of the fertilizer or manure or whatever it may be that's oftentimes high in nutrients. When I say nutrients, I'm talking about phosphorus and nitrogen primarily. So what effect do these nutrients have on water quality? Well, nutrients, particularly phosphorus and nitrogen, are building blocks of life. So our primary producers in the ecosystem require these nutrients to grow. And they are really great at growing algae. Now, algae is essential to all ecosystems, or aquatic ecosystems, and so they do provide a food source for fish, and they're kind of the building blocks of the food web in those ecosystems. But too many nutrients drive the growth of what we call harmful algae. And these algae thrive under high nutrient conditions, and they can cause a lot of issues for that body of water. So here's kind of a little graphic that I like to put up that kind of depicts a lot of the different ways that nutrients can actually enter into our rivers and streams. And eventually they may flow down into an estuary or a lake, and we see algae problems even in the ocean. I'll talk about those in a little bit. But mainly what happens is that nutrients and phosphorus will enter our bodies of water, and algae grow, these harmful algae grow really well under high nutrient conditions, and then they also need, they're also dependent on temperature and sunlight. And so usually we see them really bloom and really take over in the late summer. And so we'll get these huge blooms or these mats of algae growing on the surface or sometimes just below the surface of our surface waters. And they can cause a lot of different issues, namely one that as they die, they sink down to the bottom and then decompose. And during that decomposition process, it sucks a lot of the oxygen out of the water. And so then we'll see really low dissolved oxygen content in the water, so low that it can cause fish kills or drive them away. So it wouldn't be a sustainable place for fish to live. So some of the other issues that these algal blooms can cause, so I mentioned decreased dissolved oxygen. They can impact food availability for wildlife. A lot of times algae, if they grow in such thick mats, they can actually block out sunlight, and then emergent aquatic vegetation wouldn't be able to grow. They can cause fish kills because of the low dissolved oxygen, which is going to be a loss of economic opportunities. Lake Erie is pretty notorious for their algal blooms, and they, the one in 2011, they lost $71 million. That's mainly going to be from the fishing industry, but we also got to remember that that's ecotourism, that's people just wanting to go recreate on the lake. So it's a lot of different things kind of rolled into that value. And then finally, these blooms can actually produce a toxin, and that toxin can be directly harmful to people and wildlife. So here are some examples of kind of the different view that you might see if we get algal blooms that are growing. The bottom two pictures here are examples of non-toxic algae. So that would be kind of natural algae that would grow, wouldn't necessarily need very high nutrient concentrations in the water, but they're going to be providing food and resources for the environment. But these harmful algae up here are going to grow in these large mats. And oftentimes when people call and they're worried about harmful algae, we'll tell them that it's almost like a neon green, and it kind of also can look like spilled paint as well. So the toxin that these harmful algae can produce, I like to put this graph up just to kind of give you a sense of, you know, what we're talking about. And basically this is depicting the most toxic of these chemicals is here at the top, and then the least toxic are down at the bottom. And if you kind of scroll up and down, in the red are toxins that algae can actually produce, and these are toxins that have been that we can collect and actually test in some of our surface waters. And as you kind of look up and down this list, you might see names that you recognize like cyanide, Botox, DDT. So it really is as toxic or more toxic than some of kind of the notorious toxins that we're familiar with. These toxins have been in the news. Last year, we saw a bunch of different stories about dog deaths. One was pretty notable in North Carolina. A lot of the national news outlets picked it up, but 3 dogs had actually died for playing in a lake that had a lot of these algal growth, and the owner didn't know about the toxin, and they died very quickly afterwards. These toxins can either be pretty commonly, they're either a hepatotoxin, so they affect the liver, or they're a neurotoxin. And it can really cause, if you ingest these, a lot of times the dogs will play in the water and they'll get it on their fur, and then they'll lick their fur as they clean themselves, and they ingest it that way. And it can be several hours to maybe a day or two before they can actually have some pretty serious health effects. So I wanted to put up kind of some notable algal blooms that we see on basically an annual basis anymore. So the Gulf, the dead zone in the Gulf of Mexico, that dead zone is from very low levels of dissolved oxygen, which is caused by these huge algal blooms. And we see tens of millions of dollars lost every year in the seafood industry and tourism industry just because we're adding too many nutrients that then drive the growth of these algal blooms. Similar to the Lake Erie, I was living up there and in graduate school in 2014 when the city of Toledo actually had to issue a tap water advisory and told everybody that they shouldn't drink their tap water. You couldn't boil it to remove the toxin. Basically, you weren't allowed to use your tap water for 3 days. And so that was really impactful for that region. And it kind of really drew a light to the severity of these algal blooms. This graph down here is actually, it's a Lake Erie Severity Index referring to the severity of algal blooms for each year on Lake Erie. And what's kind of funny about this, this chart is they started doing these indices back in, I think it was 2012 or 2013, when the bloom started to get really big. And at that time, 2011 was the largest and the most severe algal bloom that we had to date. And so they based this index off of 2011. And they said, okay, well, 2011 is going to be the most severe that we're going to see. So we're going to make that 10, the top of the scale. Well, since then, we've gotten 10.5. It's even gone higher than that in recent years. And so they, were assuming it was only going to get so high, but really they've only gotten bigger. So what about rivers? I'm talking about lakes and the Gulf, but what about rivers? And that's kind of one of the questions that we wanted to answer down here in Tennessee. And I know we've got partners that are doing this around the country too. How severe is this in rivers? You think rivers, it's flowing water, and so are the nutrients or the algae really gonna congest and be able to grow, or are they just gonna flow downstream and become a problem for the Great Lakes or for the Gulf of Mexico? So we did a study last fall between September and October, and basically our goal was just to go out and collect samples, mainly myself, Dory, and then some of our AmeriCorps. We went out and collected samples from various points along the Harpeth River and its tributaries. And our goal was just to kind of do a presence absence. Do we actually see any of these toxin in the water? Previously, we have done algae surveys and worked with a professor at Austin P and identified cyanobacteria or some of these harmful algae species in the Harpeth River. And so, yeah, we found them, but are they actually producing the toxin? And do we actually need to be worried that there might be a health threat? And so we went out, we collected samples, 18, all over the place. And basically what we found is in most cases, we did not detect any toxin. But in 2 of our locations, we did collect measurable amounts of microcystin, which is one of the most common toxin produced by these algae. And the concentrations of microcystin at these 2 locations was not particularly high. But I will say that these algae, they really fluctuate in the amount of toxin that they can produce. And if we didn't collect a sample right at the correct time, or if we got it before they really started producing a lot, or got it kind of on the downswing of them producing, we may have really missed kind of the peak of toxin in the water. But really what this indicates to us is that these algae are capable of producing this toxin, and in fact, they're actively producing this toxin in certain cases. So I put down here at the bottom that the US EPA recommends that you shouldn't be in contact with microcystin in concentrations greater than 4 micrograms per liter. And so that's for just swimming and primary contact activities. But you wouldn't want it in your drinking water more than 0.3 micrograms per liter. So if you went out to one of these tributaries, and I'm sure no one's doing this, but if you're just drinking the water, then that would be pretty bad. Here's the map of the places that we actually did the sampling. So along the main stem, which is where a lot of recreators are, which is, it's good that we didn't detect any toxin there, but we were finding it in some of the smaller tributaries. And so those would be feeding the main stem. And so this is one of those issues that we as an organization are really going to kind of keep our eye on. And I think we're going to try to go out again this year and maybe do a more extensive survey and see what else we can find out about these toxin. Like I said, we only really sampled between the end of September and early October, but maybe we missed the peak when the algae are really, really growing. And so extending that time period and collecting more samples is really what we would like to do, so we can make sure that we're aware of what's going on. There was also a recent paper that was published a couple months ago that was looking at a very similar issue, and they were looking at microcystin, the same toxin, in various river systems around the country. And what these kind of shapes indicate down here, you see this microcystin, and then it's got MCYE, and saxitoxin, cylindrospermopsin, and anatoxin. Those are all different types of toxin that these harmful algae can produce. And basically what this kind of pie graph looking thing is indicating is that the algae contain the genes necessary to produce the toxin. So it doesn't mean they're necessarily actively producing the toxin, but they're able to produce the toxin. And then the triangles indicate that they actually found toxin in those places. So places like the Ohio River, we have actually, they're documented sampling in the Ohio River where they found really high quantities of toxin. I think up to like 1,000 micrograms per liter. So we were just talking about, 1, 2, 3, something like that, but over 1,000. So that's an incredible amount of toxin that can be found in some of these places. I want to talk about our pathogens. And when I say pathogens, I'm kind of referring to a wide variety of different bacteria. So here's kind of some examples of some of the pathogens that we may be interested in. And we can trace them oftentimes back to a common source. Here gives you some examples of pathways. So we might see runoff, sewage, livestock, places like that is where these pathogens can come from. And then you see some common diseases that happen. A lot of times it's gastroenteritis, stomach issues. And so it's an issue, and it can really cause kind of direct effects to human health. And so to determine if we have kind of pathogenic contamination in a water source, we really look for E. coli. And E. coli is, it's a gut bacteria. It kind of comes from the same places that a lot of these pathogens, but it's really easy to sample, and it's pretty cheap. And so that's why we look at E. coli. So this table is actually Metro Nashville, their stormwater department, and they actually go out and they do sampling on a lot of creeks around Nashville, and they're specifically looking for some water quality indicators and then also E. coli. And that's so they can monitor sewage loss or CSOs and overflow events for sewers. And if those happen, then we might see an influx of E. coli in the system. So you can see kind of some of the numbers for E. coli that they're finding. The EPA recommends that you shouldn't actually recreate or swim in water that has over 235 colony-forming units, that's what the CF uses, per 100 milliliters. This MPN number here is basically comparable to CFUs per 100 milliliters, so you can compare. When we see over 235, that's something that the EPA would not recommend you to swim in. So we do see some values that get pretty high. TDEC has a little bit lower of a standard. There's this 126. And so even more would be in violation of those two standards. And so what a lot of places around the country do is they actively sample for E. coli to indicate pathogenic contamination at recreational, whether it's beaches or rivers, or basically to help inform the public whether or not it's safe to actually be swimming or fishing or kayaking in those water sources. Traditionally, the way to do this is you walk out to the beach in the morning, you collect a sample, take it back to the lab, and then you have to grow the bacteria. And it takes about 24 hours to do that. And then you would count the colonies on that bacteria plate. And then the number of colonies you come up with would be your value for the number of E. coli in the water. The problem is that you are then reporting a value for E. coli, or you know how much bacteria is in the water 24 hours after you actually collect the sample. And so people that are going out to recreate or to swim that day wouldn't actually know if it's safe or not. And so one of the ways that we can kind of solve this issue is that we can actually develop predictive models to predict the levels of E. coli in a water body in real time. And so we'd be able to then inform the public, hey, there's we're seeing some higher levels of E. coli, or we're predicting higher levels of E. coli. And so maybe it's not safe to swim today, or maybe if you do swim, make sure you take a shower very soon afterwards, because you want to get rid of that. Wash your hands, don't ingest the water, that kind of thing. And so we can collect real-time data. Oftentimes this is water quality data and weather data, and then use those as indicators to predict the levels of E. coli that we would then expect to see in these surface waters. And so we went to do this for the Harpeth River. Oftentimes this technique is kind of done for inland beaches, or like Myrtle Beach and some oceanside beaches, but it's not really done for rivers very often. And so we wanted to do it for the Harpeth. And so we used water quality data from 3 locations that the Franklin Water Reclamation Facility actively monitors. And we collected weather data from a weather station that was very close to those Franklin monitoring locations along the main stem of the Harpeth. And so you can see some examples of some of the variables that we used. River flow, we can actually use this sign date. This is actually the day of the year. We can actually use day of the year because we usually see E. coli at higher levels in the summertime when it's warm. And this is kind of an example of how we would view the data if we were just using one variable. So turbidity indicates the cloudiness of water, and is usually a pretty good indicator of E. coli. E. coli likes to stick to sediment in the water column. And so if the water is really cloudy, we expect to see higher levels of E. coli. So what we did was we actually developed we used a bunch of different variables or indicators for E. coli. In this model here, we used turbidity, we used the day of the year, we used river flow. And this chart actually shows what our model predicted in red, and then what we actually observed the E. coli levels in black. And so you can see that the red follows the black fairly well. And when the red, we're really looking at this purple line here. And this purple line is our EPA threshold. So when we see values for E. coli that are above the purple line, that means that we would tell people, hey, maybe it's not great to swim or to be in those rivers at this time. When values are below the purple, then hey, okay, it's safe. It's safe. We don't see high levels of E. coli. And so we really want to make sure that our model is above the line when the actual values are above the line. So we're giving people the correct information. Another thing that I want to draw your attention to are these green circles. And if you notice, when the E. coli gets high enough, they actually kind of peak at this, at a little over 2,000 colony-forming units per 100 milliliters. And that's because the test that is done, the test that Franklin does, and the test that's very common done at a lot of these surface bodies or surface waters, maxes out at this value. I have seen kind of in my past work that these E. coli values can get as high as 10,000 or 15,000. But depending on kind of the test you use, you wouldn't necessarily know that. And so this just kind of indicates that, yeah, that's the value we have for that day, but it may have been much higher. We don't know. So this is a graph of that same data, but we're putting it on a linear graph so we can compare the observed values for E. coli and then what our model predicted, or the fitted values. And the goal for this graph is to get as many of the data points in either this tan section, which indicates a true positive. That means if we were to tell people it's unsafe, then it actually is unsafe. Or this blue portion down here, which is indicating, hey, we don't think that our model is not indicating that there's much E. coli, and so it turns out that there ended up not being very much E. coli. And so we can actually adjust some of these decision thresholds or regulatory thresholds and try to maximize the number of predictors or data that we're seeing in kind of our tan box or in our blue box. But for the purposes of this kind of basic study, we had an accuracy of about 70% for days that we determined that we actually predicted E. coli. And for the traditional method that I was telling you, where it takes 24 hours to actually get a value, that method oftentimes hovers around 50 to 55% accuracy. So using our predictive model, we're a lot more accurate, and we're doing it in real time. So we could tell people, or our model can actually give results that would be valuable for that same day. These other boxes would indicate a false positive or a false negative, and we can kind of minimize or maximize those depending on which one we would value more. If we're telling people that it's unsafe when really it is safe, then that's going to be a loss for recreation industry. That's a loss for ecotourism. So you don't want to do that. You don't want to kind of economically impact the region with kind of poor decisions. But at the same time, if we're having a lot of false negatives, or if we don't issue an advisory, but the water is actually unsafe, and then you're threatening human health. So there's kind of a balance that we can strike with using models like this, and that's not a question that I really know the answer to at this point, but I think it's kind of a good thing to think about, and I might ask you guys kind of what your thoughts are near the end of this. So we're working to make this model more accurate. And I'm actually working with a professor and a graduate student at Vanderbilt that are computer science engineers. And we're trying to use machine learning and decision trees to help basically make our pretty rudimentary model that we've come up with really advance it and use computer learning and machine learning to develop a really accurate model. And then hopefully we wouldn't, we can be even more accurate and be more reliant on kind of the predictions that we're getting, and really deliver a valuable product to the general public. So lastly, I'm going to kind of just touch on some of the things that Harpeth Conservancy is working on, and I tried to shorten this presentation up a little bit because I'd love to hear some feedback, or if you guys can think of ways that maybe we can collaborate with our different organizations going forward. But right now, we are pushing forward with the citizen science program. We've used a phone app called Water Reporter for the past couple years. And it's basically kind of a social media or Instagram for rivers. And people can log in and you can actually take a picture of the river or water where you're at. If you notice anything like water quality impairments, maybe you notice that the turbidity, like we just talked about, is really high, or discharges or runoff that you see flowing into a river that you don't think should be right. It's just kind of a clearinghouse where we can share all of that information and actually use it to collect data. And we've actually used that data for some of our projects. We have an Algae Watchers campaign where we're actually trying to monitor where algae is located in the Harpeth and some of the other watersheds around Tennessee. And if we see a lot of algae, then it might actually inform some of where we're collecting samples and where we're looking for these toxin. We are currently working on implementing a water quality monitoring program where we can actually get volunteer citizen scientists to go out and collect water samples. And then we can test those water samples for nutrients, for toxin, for E. coli. And ultimately, that would be really great if that water quality monitoring program can then feed into our predictive E. coli models, and we can kind of develop a whole water quality forecast for different rivers around the state. And that really help kind of tell people who want to go fish that day, they would be able to look up and say, okay, I was planning on going to this river, but it looks like maybe the bacteria levels are high. So instead, I'm going to go to another place that's on that particular day is cleaner. And so it's kind of we're working on this program now, still kind of coming up with some of the protocols and the needs and really what we want this program to look like. We've heard some really great responses from people that live near rivers, people that live on lakes that would really want to be involved and would love to kind of be one of the samplers, or that want to help with the laboratory work or help consolidate the data, that kind of stuff. So we want this data to be useful, and we want to be able to put it up on a website so people can actually see it, use it, and it's a valuable resource. And then finally, we've got some other volunteer projects. We've got a great AmeriCorps team that helps us organize river cleanups. We do tree plantings, invasive removals. I just talked about our citizen science. So if anybody's interested in this stuff, you can find it on our website. And maybe we can talk and discuss kind of the different programs and projects each of us have, and really look at kind of combining. You know, we don't necessarily, you know, we don't like to do things in a vacuum. If other people are working on stuff, it'd be great if we can collaborate and really kind of pool our resources to make a bigger impact. And I will leave it at that. And so I try to leave some extra time for questions, and if we want to kind of talk about any projects or if people have ideas, I'll let Dory chime in if she's got anything else to add. And thanks so much for inviting me to do this and listening. This was really great.
Hey, Ryan. Ryan, this is Terry. And thank you for that great presentation. It was really great, very informative. Dory, I wanted to just kind of turn it over to you, see if there's anything you'd like to add.
Hi. Yeah, not much. I mean, Ryan was talking about our science program, and some of it's tied to the work we've been doing for years. Be familiar, because some of you are participating in a nutrient enrichment TMDL that's being designed for the Harpeth that the EPA is working on. Ryan will be trained on how to use those fancy WASP computer models. So, you know, we will be housing some of that expertise that we can then share with y'all as we try and do some of this in other parts of the state. And one of the things we're trying to figure out, there's a lot of agencies involved, TDEC and the cities and other players, Metro, others collecting data on the Harpeth, is how to do the predictions to prevent conditions for toxic algae blooms. And that's, you know, that's why we're sort of, it's a bit of a trick because some of the models don't have all the data they need. So this is obviously an evolving process. But the other thing he was discussing that we were really interested in is that some of you have been around a while, you know that a long time ago, TDEC, I think, funded sort of, hey, what are some of the water quality citizen volunteers, water quality sampling programs that Pennsylvania and other states use. And Ryan has a lot of background in that. And as he was talking about it, I realized, you know, we ought to just dive in and start designing something. And he feels that E. coli is the place to start because it's sort of the classic public health indicator. So I thought that'd be really helpful for y'all to sort of see where we are, and then we could explore. I know Tennessee Wildlife Federation has people do a lot of fishing and interest with water quality on big lakes. So maybe there's some things that we can combine together. So this was sort of like to at least give you a sense of what we've been doing.
Great. Well, I'll just open it up for questions for Ryan or Dory.
Maybe you weren't ready for math on lunch break.
Yeah, sorry about that.
Any questions out there?
Hi, I have a question. Can y'all hear me?
Yeah.
Yep.
Great. Thank you so much. This was fascinating. I know you mentioned it, and I must have missed it. Where do y'all take your samples of the river?
So we've done kind of some, I guess I'll say like event studies. So last year we kind of sampled all over the watershed, and we've done some algae sampling in the past, but really we're kind of hamstrung by consistent long-term monitoring. And that's hard to come by. We've funded a bunch of this in the past with USGS stream gauging stations. So there was one on Highway 100. one on Cotton Lane. There's 5 or 6 around the main stem of the Harpeth. The data that I was talking about, the E. coli data, is really the only consistent E. coli data along the Harpeth. And that is just upstream of the city of Franklin, just downstream of the city of Franklin, and at, I think Cotton Lane is the 3rd spot they do it. But kind of having consistent E. coli sampling is really what we need for these types of predictive models and these types of kind of to inform the public, and it's really not done anywhere else.
And that's a really good question, because this is where you get into all these intersections with policy. You have a city like Franklin, and I'm sure Metro, Nashville, and some other utilities probably have some sampling requirements in their sewer permits. And so, is the data being collected in a way that's valuable for any interpretation? Are there maybe potential opportunities for some basic data to be collected by other permittees? In the Harpeth, we coordinated, and it wasn't just our organization, it was EPA, City of Franklin, TDEC, and everybody put together a monitoring plan for a couple of years, sampling plan for the nutrient reduction strategy stuff. But there's no long-term monitoring established yet. And so that sort of begged this whole question of how do you know if any of those plans that might come out of the model actually achieve the water quality improvements you're looking for?
Right. That's interesting. Yeah. And like you're saying, bringing it to a level where it would be useful information for government entities to, I would hope at some point any huge discrepancy between what y'all are predicting and what the samples are showing would show government entities if there's any nefarious activities going on, et cetera, too. It would be really fascinating.
Yeah, that's a great point.
And that's a good point. Yeah, and that was one of the reasons. Yeah, that's one of the original out of Chesapeake Bay, and so it's one of the opportunities with is people can take a picture of something. It may turn out to be nothing, but it gives all of us an opportunity to see if there's an issue and then go try and solve it and find out if there's a recurring problem.
Right.
I jumped in on Ryan if you want to add anything else.
Oh, I was just going to say that that's kind of another reason for establishing our kind of this water quality monitoring program that we want to get going, because you do really need these long-term regular monitoring programs to know if you're seeing, if you see a spike in, pollution or contaminants or whatever, is that an anomaly? Is someone doing something that they shouldn't, and then oftentimes with some of these, you can actually then like pluck out a sampling site and say, okay, well, we're seeing really high E. coli, for example, at this site, every month or whatever. We can actually do some data analysis and do what's called source tracking studies to figure out if that E. coli is coming from livestock, is it coming from a human source, is it, and so you can actually then kind of try to isolate or kind of figure out the source of it. And maybe it is someone doing nefarious activities, but that's something that we would want to know. And right now we wouldn't know.
And as far as some of the costs for sampling, like some of you all with your membership and people and partners across the state, I'm thinking of the Wolf River Conservancy and others. If you have some areas you'd like to see sampled on a regular basis, we're not launching this live or anything soon. We would need some places to start working and developing the predictive models. We'll be working with Department of Health and TDEC and others as well, and going over protocols and things like that. But we know we need some locations in the Harpeth because of the work we've been doing for so long, but we want other locations in the greater Nashville region as almost like, what do you call it, like beta testing? Mm-hmm. And some of the hard costs for some of the lab costs isn't that tremendous at all. A lot of the time is Ryan, designing all of the work behind the scenes and working with the agencies. Ryan, you wanted to mention something about some of the data processing though. Like if we were interested in working with Wolf River, we might need a local location to run some of the data, because we have an arrangement right now set up with here in the Middle Tennessee area, but samples in West Tennessee would be different.
Yeah, some of the logistics would, we'd have to work out, but it's, E. coli, kind of a lot of them basic water quality parameters that we're interested in, or that we would want to be able to kind of feed into any kind of predictive model or whatever. I mean, can be done at a variety of different labs, you know, so it's like maybe we would find a local partner that would help analyze those samples, or, you know, maybe we're shipping them to the lab that we use. And, you know, I've done that in the past and that's not a problem at all. I used to have to send samples to Denver, you know, from Ohio to get analyzed. So it's really not that big of a deal. And I guess the other thing I'd like to mention too is when I talk about this kind of like predictive model and like advising the public that it's unsafe to swim or to be in the river, I know that's like kind of sounds scary, but it is done all over the US and various beaches, and this is already kind of being done, these types of programs. And we are already we're talking with TDEC and kind of like the language or how to do this, we haven't quite figured out, but we definitely want to do it in a way that people are okay with it, and that the state's okay with it, and the other organizations, you know, that we're not, you know, giving people information or like being like whistleblowers unnecessarily or anything like that. Like this just needs to be, we really think it could be a really valuable tool, you know, not that it needs to like shine light on something that people are, you know, that, you know, us saying it in like a bad way, using verbs that, you know, really kind of cause panic. Like that's not the way that like this needs to be developed and used.
Hey, Ryan, you mentioned sort of the app for some of the citizen science stuff.
Yeah.
Is that on your site? Does that run on Android and Apple? Or is that a sort of your all's app, or is that more of a universal app?
No, it's a universal app. It's all across the nation. We are kind of we're like the administrators for it in Tennessee. But you can get it from your app store. You can also Google Water Reporter, or you can go to our website. We've got kind of a how-to sign up to get on it. And then, yeah, and then you can just kind of start posting things and we can see it, you can share them. Yeah, so it is a really great resource.
Okay, thank you.
Yeah, so if some of y'all would like to push it out there right now in Tennessee, do we just have one group in Tennessee? It's like you join the Harpeth Conservancy group and you'll see all the posts across the state of Tennessee. Then we can set up some other administrators so that some of y'all can actually get the feedback, or Ryan can get it and send it to you. But it makes maps too, so you automatically know where the location is and what's being reported immediately. And so if there is something that, you know, like it's an oil sheen, someone can actually go respond to it.
Yeah, we have had some really great success with people posting things on it and say that there's a, you know, I think last summer we had someone that posted to it that they found someone had dumped a whole bunch of manure just straight into the river. And so we got that information passed on to the appropriate people like within an hour of them posting it, you know, and so it was really great and a quick response.
Yeah, have the agencies been pretty responsive to it?
Yeah, no, they've been really great whenever I've kind of forwarded them stuff, or when we've found things that you know, that are problematic. They've been really quick to respond and have been thankful for, you know, bringing that to their attention.
And usually it's either us or it'd be another, you know, local partner too that might be the right player to actually talk to that property owner or things like that. You know, so you're not trying to, what do you play gotcha. But I think there's a lot of people who don't realize that, you know, they might be managing a horse farm or they just have a chicken coop and it's like, it wasn't a really cool idea to dump it in the creek. It just, you know, that kind of thing.
Any other questions for Ryan or Dory? So Ryan, we really want to thank you. It was a great presentation. Just so you know, we are starting to record these now. So as a follow-up, you know, the bat work in cave and karst, and this one too, we'll send out links so people who could not make it would like to view it again and get some of that information. And please reach out to Ryan or Dory if you have some follow-up questions or some partner ideas or need some additional information. We really want to help people kind of reaching out. You got Ryan's contact information up there. So thank you. Next week, we have another brown bag on Wednesday at noon. The Tennessee Wildlife Federation will be discussing some of their focus on Asian carp.
Oh, perfect.
We will be sending out that link and a reminder next week again. And so please continue to just join us and learn together. So hope everyone's well and safe, and we look forward to joining you next week.
Thanks so much.
Okay.